You carry the remains of a war inside your body. Roughly 8% of your DNA is viral code, fragments of ancient retroviruses that infected your ancestors and never left. This viral graveyard takes up 4 times more space than the genes that code for human proteins, which occupy only about 1 to 2% of the genome. Most of these sequences are molecular fossils, broken and silent, but one of them tells a story that rewrites what we think a pandemic can be. Between 33 and 15 million years ago, a retrovirus called ERV-Fc swept across the planet. It didn't burn through populations in months or years. It persisted for 18 million years, longer than the entire history of the human genus. It jumped species at least 20 times, infecting the ancestors of dogs, bears, primates, rodents, and dozens of other mammals. Then it vanished, leaving only its genetic signature embedded in the genomes of its hosts. The virus is extinct, but we can still read its biography, written in the language of DNA. Pandemics, as we understand them, are historical events. The Black Death killed millions in the 14th century and disappeared. The 1918 influenza pandemic burned through the global population in 2 years. COVID-19 emerged in 2019 and, within 3 years, had transitioned to endemic circulation. These outbreaks operate on human timescales, years, decades at most. ERV-Fc operated on geological timescales. It emerged when Antarctica was freezing over, when the first grasslands were spreading across continents previously covered in jungle. The ancestors of modern humans wouldn't appear for another 13 million years after it went extinct. The virus actively jumped between mammalian orders, from carnivores to primates, from primates to rodents, from rodents back to carnivores. It spread across every continent except Australia and Antarctica. And then, around 15 million years ago, it stopped. The infectious form went extinct. What remains are molecular fossils, fragments of viral DNA embedded in the genomes of 28 modern mammal species, including us. These fragments are broken, mutated beyond function, but readable. This is the story of the longest pandemic in mammalian history, reconstructed from the viral graveyard inside our cells. The human genome contains roughly 3 billion base pairs, the chemical letters that encode biological instructions. Only about 1 to 2% of those letters code for proteins, the molecules that build tissues and run cellular machinery. The rest was once dismissed as junk DNA, evolutionary debris with no clear function. That dismissal was premature. Tucked into the non-coding regions are hundreds of thousands of genetic sequences that resemble viruses. They carry recognizable viral genes, instructions for building capsids, enzymes for copying RNA into DNA, proteins for fusing with cell membranes. But they're broken. Mutations have shattered the original code, turning functional genes into genetic wreckage. In 2016, a team led by William Deal and Welkin Johnson at Boston College scanned the genomes of 50 mammal species, searching for traces of ancient retroviruses. They found fragments of one particular virus, ERV-Fc, in 28 of them. Humans, chimpanzees, gorillas, baboons, macaques, squirrel monkeys, dogs, pandas, ferrets, mongooses, spotted hyenas, mice, rats, squirrels, and even the aardvark. The distribution was global. ERV-Fc appeared on every continent except Australia and Antarctica. Australia had separated from other land masses before the virus emerged, isolating its marsupial fauna. Antarctica was frozen and uninhabitable. Everywhere else, the virus found hosts. The researchers applied molecular clock techniques to date the insertions. The oldest traces appeared approximately 33 million years ago in the early Oligocene epoch. The youngest dated to around 15 million years ago in the Middle Miocene. No insertions appeared after that point. The virus went extinct as an infectious agent. The only traces that remain are fossilized proviruses locked in mammalian genomes. 18 million years of continuous activity. That makes ERV-Fc the longest documented pandemic in mammalian history. Viruses have no bones. They're molecular machines, strands of genetic material wrapped in protein shells. When a virus dies, it decomposes completely. No fossil forms in rock. Retroviruses, however, leave a different kind of record. They replicate by inserting a DNA copy of their genome into the host's chromosomes. Usually, this happens in somatic cells, lung tissue, liver, blood. The viral DNA dies when the host dies. But if the virus infects a germline cell, a sperm or egg, the insertion becomes heritable. Every cell in the resulting offspring carries the viral sequence. If that offspring reproduces, the sequence spreads through the population. The host genome becomes a fossil bed, preserving viral sequences across geological timescales. The 2016 study by Deal and colleagues used computational methods to mine these fossil beds. They started with the known genetic structure of modern gamma retroviruses, simple viruses like murine leukemia virus and feline leukemia virus. These viruses share a basic architecture, 3 core genes called GAG, POL, and ANV. GAG encodes the structural proteins that form the viral core. POL encodes reverse transcriptase, the enzyme that converts viral RNA into DNA. ENV encodes the envelope protein, which allows the virus to fuse with host cell membranes. The researchers wrote algorithms to scan mammalian genomes for sequences resembling these 3 genes. They searched public databases containing the genetic blueprints of 50 species. 28 species contained sequences that matched the structure of a gamma retrovirus with high confidence. The sequences were degraded. Millions of years of random mutation had introduced errors, insertions, deletions, substitutions. But by comparing the damaged sequences across multiple species, the researchers could reconstruct the original viral genome through consensus. If most species had the same nucleotide at a particular position, the original virus likely carried that sequence. This triangulation allowed the team to rebuild the sequence of ERV-Fc as it existed before it went extinct. They assembled the molecular architecture of a pathogen that had been gone for 15 million years from millions of scattered genetic fragments. A retrovirus carries its genetic information as RNA, not DNA. When it infects a cell, it converts that RNA into DNA using an enzyme called reverse transcriptase. The viral DNA then integrates into one of the host's chromosomes, becoming a permanent part of the host's genetic blueprint called a provirus. The integrated provirus hijacks the host's cellular machinery. The cell reads the viral genes and manufactures viral proteins. These proteins assemble into new virus particles, which bud off the cell membrane and infect neighboring cells. Most retroviruses infect somatic tissues and die with the host. HIV, for example, targets immune cells and spreads through bodily fluids. When the host dies, the infection ends. ERV-Fc followed a different path. At some point during its 18 million year reign, it infected germline cells in multiple mammalian species. Germline infections are rare. Sperm and egg cells are sequestered in reproductive organs, protected from most pathogens. The virus must not only reach these cells, but integrate into their DNA without killing them or preventing fertilization. The infected gamete must then participate in successful reproduction. When a germline cell carrying an integrated provirus fertilizes or is fertilized, the resulting offspring inherits the viral DNA in every cell of its body, including its own germ cells. If that offspring reproduces, the provirus spreads further. Over generations, the viral sequence can become fixed in the population, present in every individual of the species. This process, called endogenization, turns a transient infection into a permanent genetic element. The virus stops being an infection and becomes a gene. Across millions of years and billions of animals, the statistically improbable becomes inevitable. ERV-Fc achieved germline insertion dozens of times across dozens of species. Once trapped in the host genome, the provirus mutates at the same rate as the host's DNA. Random errors accumulate. Stop codons appear in the middle of viral genes. Regulatory sequences degrade. Eventually, the provirus becomes defective, unable to produce infectious particles. The virus is genetically dead, but its sequence remains as a record of its existence. Dating ancient viral insertions requires molecular clocks. When a retrovirus integrates into a host genome, it carries two identical sequences called long terminal repeats, or LTRS, at either end of its genome. At the moment of insertion, the left LTR and right LTR are perfect copies of each other. After integration, the two LTRS mutate independently. Random errors accumulate in each copy at the rate governed by the host's DNA replication machinery. By counting the number of differences between the left and right LTRs, and knowing the mutation rate of the host genome, researchers can calculate how long the provirus has been sitting in the DNA. The Deal study applied this technique to ERV-Fc insertions across multiple species. The oldest insertions, found in lineages that diverged early in mammalian evolution, dated to approximately 33 million years ago, the early Oligocene epoch. The youngest insertions dated to around 15 million years ago in the Middle Miocene. The timeline can also be constrained by the evolutionary relationships of the host species. If two species both carry ERV-Fc in the same genomic location, meaning the insertion occurred before they diverged from a common ancestor, the insertion must predate the split. Paleontologists know from fossil evidence when many mammalian lineages diverged. Old World monkeys and apes split roughly 25 to 30 million years ago. If both groups carry the same ERV-Fc insertion at the same chromosomal position, the infection happened before that split, in their shared ancestor. By cross-referencing LTR divergence with host phylogeny, the researchers built a detailed chronology of ERV-Fc's spread. The virus emerged in the Oligocene, reached peak activity in the early Miocene around 23 to 20 million years ago, and disappeared in the Middle Miocene, 18 million years from emergence to extinction. The Oligocene began 33.9 million years ago with a sudden climate shift. The warm, humid greenhouse of the Eocene collapsed. Antarctica froze over, locking vast amounts of water into ice sheets. Sea levels dropped, global temperatures fell. Tropical rainforests, which had stretched across much of the planet, retreated toward the equator. In their place, open woodlands and the first grasslands began to spread. These new ecosystems offered longer sightlines and fewer hiding places, favoring fast-moving herbivores and cursorial predators. The fauna of this world looked strange but recognizable. Giant hyenodonts, carnivorous creodonts the size of modern bears, hunted through the woodlands. Early saber-toothed cats called nimravids stalked prey with blade-like canines. Primitive monkeys moved through the trees in Africa and Eurasia, the first anthropoid primates to venture beyond tropical forests. The ancestors of modern carnivore families, early dogs, bears, and weasels, were small but rapidly adapting to the open landscapes. Mammals diversified rapidly into these new niches. Anthropoid primates spread from Africa into Eurasia. Early carnivores radiated into the expanding woodlands. Rodents, small and prolific, filled niches from forests to grasslands. The cooling climate and opening landscapes created ecological opportunities absent in the dense Eocene jungles. The Miocene, beginning 23 million years ago, amplified these trends. Grasslands expanded further, creating savannas and steppes that covered entire continents. This was the Age of Grass, a time when open landscapes replaced closed forests across much of the globe. The visual transformation was dramatic. Where dense canopy had once blocked the sky, endless seas of grass now stretched to the horizon. Tectonic activity was reshaping global geography. The Tethys Sea, which had separated Africa from Eurasia for tens of millions of years, gradually closed as the African plate collided with the Eurasian plate. The collision proceeded slowly, but relentlessly. Shallow seaways gave way to marshes, then dry land. By the early Miocene, around 20 million years ago, animals could walk between continents that had been isolated since the age of dinosaurs. In the north, the Bering Land Bridge connected Asia and North America intermittently as sea levels fluctuated with glacial cycles. When sea levels dropped, a broad plain emerged between Siberia and Alaska. Mammals crossed between the Old World and the New World, carrying parasites and pathogens with them. Populations that had been geographically isolated for millions of years suddenly came into contact. A primate species that evolved in Africa could now encounter carnivores that evolved in Europe. Rodents from Asia could spread into African ecosystems. The biogeographic barriers that had kept these lineages separate dissolved. Viruses that had circulated in isolated populations for millions of years suddenly had access to naive hosts, animals with no evolutionary history of exposure, no immune adaptations to defend against the pathogen. A virus adapted to African primates could jump to a Eurasian carnivore that had never encountered anything like it. If the virus could bind to the carnivore's cell receptors, if it could replicate in the carnivore's tissues, transmission succeeded. ERV-Fc exploited these opportunities. The virus spread across continents, infecting diverse mammalian lineages as they encountered one another in the changing landscapes of the Oligocene and Miocene. The geographic range reconstructed from modern genomes spans Africa, Europe, Asia, and North America. Only Australia, already separated by ocean, and Antarctica, locked in ice, escaped infection. Most viruses are specialists. They evolve to exploit a narrow range of hosts, binding to specific cell surface receptors that exist only in closely related species. A virus that infects bats typically cannot infect humans unless it undergoes significant evolutionary adaptation. ERV-Fc was a generalist. The Deal study revealed something unexpected when they reconstructed the viral phylogeny, the evolutionary tree built from ERV-Fc sequences. If the virus had simply infected a common ancestor and then diverged passively as host lineages split, the viral tree and the host tree would be identical. A virus in dogs and a virus in bears would be closely related because dogs and bears share a recent common ancestor. A virus in primates would be distantly related to both. The pattern was different. Viral strains found in carnivores were nearly identical to strains in primates. Rodent strains appeared nested within carnivore lineages. The trees did not match. The most parsimonious explanation was that the virus jumped between species at least 20 times during its 18 million year existence. The host list from the 2016 study reveals the breadth of transmission among carnivores, dogs, pandas, ferrets, mongooses, and spotted hyenas. Among primates, humans, chimpanzees, gorillas, baboons, macaques, and squirrel monkeys. Among rodents, mice, rats, and squirrels. The virus even reached the aardvark, a member of Afrotheria, an ancient mammalian lineage. Cross-species transmission requires close contact. Predation and scavenging are primary mechanisms. A carnivore that kills and consumes an infected primate could be exposed to high concentrations of the virus in blood and tissues. A scavenger feeding on an infected carcass encounters the pathogen in organs and bone marrow. If the virus can bind to the carnivore's cell receptors, infection follows. The virus enters through the digestive tract, crosses into the bloodstream, and begins replicating in the new host. Shared water sources, overlapping territories, and direct physical contact in mixed species feeding sites all create opportunities for transmission. In the open grasslands of the Miocene, animals congregated at rivers and waterholes. Predators and prey came into proximity. Herbivores grazed alongside one another. The ecological mixing created viral opportunities. The expansion of grasslands in the Miocene likely increased these interactions. Open savannas bring herbivores, predators, and scavengers into proximity. Rodents, which thrive in grassland ecosystems and appear frequently in the ERV-Fc host list, may have maintained circulating viral populations that spilled over into larger mammals through predation. A mongoose hunting infected mice could contract the virus. A hyena scavenging an infected mongoose carcass could contract it again. The virus moved through the food web. Carnivores show diverse ERV-Fc strains in the genetic data. Primates and rodents, which often occupy overlapping habitats, and are preyed upon by carnivores, show viral strains that appear in carnivore lineages. The virus exploited the ecological relationships between predators, prey, and scavengers. The virus also recombined. When two different strains of ERV-Fc infected the same host cell simultaneously, their genomes could swap segments during replication. The resulting hybrid viruses carried genes from both parent strains, mixing traits that had evolved in separate lineages. The Deal study found clear evidence of this genetic shuffling in the fossil record. Viral strains showed mosaic patterns: the GAG gene from one lineage, the ENV gene from another. Recombination allowed ERV-Fc to rapidly change its surface proteins, evading host defenses and adapting to new species. A virus that evolved in rodents could acquire an envelope protein from a carnivore virus, giving it the ability to infect primate cells. The genetic mixing accelerated viral evolution and expanded host range. Mammals did not passively endure ERV-Fc. They evolved defenses. Retroviruses are vulnerable at multiple stages of their life cycle. After entering the cell, the viral capsid, the protein shell surrounding the viral RNA, must remain intact long enough to deliver its cargo to the nucleus. Mammals evolved a protein called TRIM5-alpha that recognizes the capsid structure and disassembles it prematurely. The viral RNA is released into the cytoplasm, where it is degraded before reverse transcription can occur. Another defense targets reverse transcription itself. The protein ApoBec3 binds to viral RNA and introduces mutations during the synthesis of the DNA copy. It hypermutates the viral genome, scrambling the genetic code so that the resulting provirus is non-functional. The virus integrates into the host chromosome, but the genes are nonsense. A third weapon prevents newly assembled viruses from leaving the infected cell. Tetherin, also called BST2, acts as a molecular anchor. It embeds in the cell membrane and in the viral envelope simultaneously, physically holding the budding virus in place. The virus cannot detach and spread to neighboring cells. These defenses are not universal. They evolved independently in different mammalian lineages, shaped by the specific pathogens each lineage encountered. The TRIM5-alpha variant in humans differs from the variant in Old World monkeys and carnivores. Each version is adapted to recognize the capsid structure of the retroviruses that infected that lineage's ancestors. ERV-Fc drove this evolutionary arms race. As the virus evolved to evade one defense, mammals evolved a counter-adaptation. This coevolutionary feedback loop forces both parties to continually innovate. A mutation that allows the virus to partially evade TRIM5-alpha gives it a selective advantage. Hosts with a slightly more effective TRIM5-alpha survive better. The cycle repeats across millions of years. Modern humans use these ancient weapons against contemporary threats. TRIM5-alpha defends against HIV, recognizing and dismantling the viral capsid before infection can establish. ApoBec3 targets a wide range of retroviruses, introducing lethal mutations that destroy viral genomes. Tetherin restricts viral budding in cells infected with multiple types of pathogens. These proteins are molecular scars, evolutionary memories of a pandemic that ended before the first hominins appeared. The fact that ERV-Fc eventually went extinct around 15 million years ago suggests several possibilities. The virus may have saturated its host populations, running out of susceptible individuals. Mammalian immune systems may have evolved a broadly effective defense that the virus could not overcome. Perhaps a critical mutation in a cell surface receptor that rendered multiple species resistant simultaneously. Alternatively, climate disruption during the mid-Miocene cooling event may have driven reservoir species to extinction, cutting off the source of new infections. The research does not definitively resolve which scenario occurred or whether multiple factors combined. ERV-Fc is extinct as an infectious virus. No modern mammal harbors a functional, replication-competent copy. The viral sequences that remain in mammalian genomes are defective. Mutations have introduced stop codons, deleted essential genes, and scrambled regulatory elements. The proviruses cannot produce infectious particles. Extinction, however, is not erasure. The viral DNA persists, copied into every descendant of the original infected individuals. The fossilized proviruses occupy fixed locations in the genomes of their hosts. Humans carry ERV-Fc fragments on specific chromosomes. So do chimpanzees, gorillas, baboons, and macaques, each species carrying a slightly different set of insertions, reflecting the independent history of infection in each lineage. Over millions of years, some endogenous retroviruses have been co-opted by their hosts. The process, called exaptation, occurs when a viral gene mutates into a form that benefits the host rather than the virus. The most famous example is syncytin, a protein originally encoded by a retroviral ENV gene. Mammals use syncytin to fuse placental cells, creating the barrier between maternal and fetal blood supplies. Without syncytin, placental mammals could not exist. The research suggests hints that some ERV-Fc sequences might play regulatory roles. A few fragments lie near genes involved in immune function and development. They could potentially act as genetic switches that control when and where nearby genes are expressed. If a viral promoter happens to activate a useful gene in the right tissue at the right time, natural selection can preserve it. The evidence for ERV-Fc domestication remains speculative, but the principle is established in other endogenous retroviruses. The distinction between host and virus blurs over deep time. An invader that enters the germline ceases to be foreign in the conventional sense. It becomes part of the genetic architecture, subject to the same evolutionary forces as any other sequence. If it confers a fitness advantage, it spreads. If it imposes a cost, it degrades. Most ERV-Fc sequences have degraded into genetic noise. A few may have been repurposed into functional elements of the mammalian genome. The pandemic did not resolve in the traditional sense. There was no final battle, no moment when the last infected animal died and the virus disappeared. Instead, the virus transformed. It stopped replicating, stopped spreading, stopped being a pathogen. It became information, a record of an ancient infection frozen in the genome and passed down through hundreds of thousands of generations. Every human carries fragments of ERV-Fc in their DNA. So do chimpanzees, gorillas, and dozens of other species. The virus that swept across the Oligocene and Miocene worlds, that jumped between carnivores and primates and rodents, that persisted for 18 million years through tectonic upheavals and climate shifts, is gone. The infectious form vanished 15 million years ago. What remains is a molecular fossil, a genetic scar from the longest pandemic in mammalian history, preserved in the genomes of its survivors.